So I welcome you all to the first talk of this workshop. So I hope you all had your morning coffee and you are all pumped up to actually learn some basic molecular biology. So this will be a nice jog up kind of a lecture for students from biology background. And for the participant who do not have a biology background, so during this lecture, you're going to hear few keywords which will basically help you in the coming 2 days of this conference. So there is a great lineup of speakers and we all are looking forward to hear some of the good talks in the field. So let us begin with learning some fundamentals. So I'll start with the fundamental unit of living system, which we all know is a cell. So there is a diversity in the cell types. There are two major types of cells, one is the prokaryotic cell type, the other one is the eukaryotic cell type. So prokaryotic cell type, basically the example is bacteria. And for eukaryotic cell type, the cells which are found in human body are the eukaryotic cell types. The main difference between the two is that the genetic material, which is called the DNA, in case of prokaryotes, it is present inside the cytoplasm, whereas in case of eukaryotes, it is present inside a membrane-bound organelle, which is called the nucleus. There is, of course, differences in the way the DNA is packed. The DNA in prokaryotes is circular, whereas in eukaryotes, it's linear, and it is condensed with the help of packing protein into further there are different levels of packing of the DNA in the eukaryotes. There are certain membrane-bound organelles present in the eukaryotes, like mitochondria, endoplasmic reticulum, which are absent in prokaryotes. So now I mentioned that DNA is the genetic information storehouse for the cell. So this just represents how DNA is packed inside the eukaryotic cell. So in eukaryotic cell, there is nucleus. Inside the nucleus are present chromosomes. So chromosomes are made up of this genetic material called the DNA. Now let us start by looking at what is DNA and what it is made up of. So DNA has this double helix. It is around 2 nanometers in size, the diameter of this double helix. If we look at the basic unit by which it is made up of, so it is made up of these nucleotides. So a nucleotide has 3 main parts. One is the sugar, which is a 5-carbon sugar called the ribose sugar. In case of DNA, on the second carbon, so this is the first carbon and this is the second carbon, instead of a hydroxyl group, there is just a hydrogen atom. So there is 1 oxygen less. So that is why it is called deoxyribonucleic acid. On the first carbon is attached covalently bond a nitrogenous base. Nitrogenous bases also falls into 2 categories, purines and pyrimidines. So purines has this double ring-like structures. So purines include adenine and guanine, whereas pyrimidines have cytosine and thymine. And pyrimidines has a single ring-like structure. So if you if somebody has handled the DNA or the nucleic acid for that matter, they would remember that it absorbs light at 260 nanometer. So this is mostly because of the nitrogenous base present in the structure of the DNA that is responsible for that absorption. Now on the 5th carbon, there is present a phosphate group. This phosphate group is involved in the linkage with the next nucleotide. So using those phosphodiester linkages, a sort of backbone of DNA double helix is formed, and the nitrogenous bases are projected in between that double helix, and these nitrogenous bases are involved in the hydrogen bonding. So hydrogen bonding is a non-covalent type of interaction. It plays a very important role in holding a lot of biological molecules, DNA being one of them. So there are certain rules by which this DNA is organized. So if you remember that there is a lot of contribution from different fields. So at the end, Watson and Crick in 1950s, they got Nobel Prize for elucidating the structure of DNA, but they had huge help from Maurice Wilkins and Rosalind Franklin, who basically identified the X-ray crystal structure of DNA, and the char graphs rules basically define what combinations these bases interact with each other. So that that forms the basis of this particular structure that we know now today. So A adenine and T thymine, they are involved. There is a double hydrogen bond, in, which is by which they are interacting with each other, whereas guanine is basically involved in interaction with cytosine with the help of 3 hydrogen bonds. Now there is a polarity also involved with the nucleic acid. So they have a direction. So they have a 5' end which has a free phosphate group, and they have a 3' end which has a free hydroxyl group. The double helix is anti-parallel. So one strand is running in 5 to 3' direction and the other one in the opposite direction. So by convention, we read DNA string in the direction of the transcription. Transcription basically happens from 5' to 3' direction. So you must have seen the way we write the nucleic acid. So we say, for instance, in this example, at the 5' end, there is a cytosine. So we say CAGT. So this is 5' to 3' direction. And for the other strand, the 5' to 3' direction is in this in the opposite way. So there is GTCA. Now the DNA in eukaryotes is condensed to actually fit into that particular nucleus. So as I mentioned that the double helix, the diameter is around 2 nanometer. It packs around these proteins called histones and forms this bead on a string-like structure, which is called the nucleosome, and it's around 11 nanometers in diameters. Then this nucleosome further condenses with the help of some scaffold proteins and forms this chromatin structure, which is around 30 nanometers. This chromatin structure further undergoes different levels of condensation. It loops out and it finally forms this 1.4 micron structure, which is called the chromosome. So you might have seen these chromosomes if you have done some basic biology experiments. For example, during the cell division, you see that during the metaphase stage of the cell cycle, these chromosomes are arranged at the metaphase plate. Now, talking about chromosomes, so every organism has a sort of fixed number of chromosomes. So in humans, we have 22 pairs of autosome and 2 pairs and a pair of sex chromosome. So in somatic cells, which are non-reproductive cells, so you have these 22 pairs. 22 pairs, these are called homologous chromosome. Every pair, so this is a chromosome number 1. These are homologous chromosomes. One is from the mother and one is from the father. So you have 22 such pairs, plus depending if you are female, you have a XX chromosome, or if you are male, you will have a XY chromosome. So 23 into 2, so in total there will be 46 chromosomes in the somatic cells. But the germline cells in humans, they are haploid. They will not have this, this is a diploid phenotype, but these are haploid. So they will have half of the number of chromosomes. So 22 autosomes and 1 sex chromosome. So there will be total 23 chromosomes in the germ cells. Now the DNA that I mentioned, I said that it is the genetic storehouse of the cell. So it has all the information how the cell is supposed to be behaving, but it itself is not doing the work for it. So what the DNA does is that it basically creates some workhorses. which are called the proteins, which can basically do the work for DNA. Now there are different, there is an intermediate molecule which is called the mRNA or messenger RNA, which is a mediator that basically, whatever information that is present in the DNA, that is translated and a protein is formed. So this is also the central dogma of molecular biology that we know now. So in this central dogma, there are certain important steps. One is called the transcription. So in the process of transcription, so the DNA has all the genetic information. There are certain promoter sequences. There are certain protein coding sequences, which is depicted in blue in this picture. There are certain, and these protein coding regions are called the exons. There are certain sort of intersecting or the non-protein coding regions, which are called the introns. So initially people thought that the introns were like junk DNA because they are not coding for any protein. But now we all know that introns also play a very important role in the regulation of gene. So they are not there just like that. There is a purpose that we have introns in our genetic material. There is always a start site. Some sequences are important for the transcription starting, and at the end of the gene, there will be a stop site. So RNA polymerase will recognize the promoter, will sit on this DNA, and it is going to read these codes which are present in the DNA, and it is going to transcribe it with a process called the transcription. I'm not going into too much detail. It's an overview. So you will now a sort of mRNA, messenger RNA, which is also called the transcript, is going to be formed. Now the transcript has the information for protein coding, which is interrupted by the information which does not code for the protein. So there is a requirement to splice off these intron non-protein coding region from the mRNA. So this transcript or the messenger RNA undergoes splicing event. That's also a highly regulated event, and it is also important for the gene regulation. So the introns are spliced off and the exons are joined together, and this is the final finished product that you will get, a continuous information of protein coding sequences. This is the mature messenger RNA, and that basically is read by the translational machinery, and a protein is formed. The protein, once formed, will also undergo a lot of post-translational modifications, for instance, phosphorylations or any other kind of post-translation modification that will basically help in the folding of the protein as well as regulate the function of the protein. And finally, we'll have this sort of workhorse for the cell. And this is the finished protein that is going to go and do the function for that particular cell. So now I have introduced you to this another nucleic acid, which is called the RNA. So let us look at the RNA and how it is different from the DNA. So RNA is ribonucleic acid. So the major difference from the DNA is that it is a sort of not a double helix, but unlike the DNA, it's like a single standard molecule. One important difference in terms of the nitrogenous bases is that instead of thymine, it has a uracil. So adenine can bond with uracil in this case of RNA. The RNA also forms secondary structure. So you will see that they have a sort of stem loop-like kind of structure. They also have a polarity 5' end that has a free phosphate group, and the 3' end which has a free hydroxyl group. There are different types of RNA molecules. These are major types. This is a messenger RNA. So basically it gets translated. It has the information which is read and to make the protein. Then there are ribosomal RNA. So ribosomal RNA basically help in the translation. They together with they associate with certain proteins and they form these ribosome subunits. And these ribosomes basically assist in reading this mRNA. It helps in translation. Then there is transfer RNA. Transfer RNA helps. It has two ends basically. One end is recognizing the code which is present on the mRNA, reading it, and the other end is basically trying to catch the particular amino acid which is suitable for that particular code which is present in the mRNA. Now I'm talking about this genetic codes. So genetic codes, it's amazing the kind of information present in the DNA. So there is a sort of code. The information is not like random. You have certain rules or guidelines which is followed by this molecular machinery. And this is a great amount of work that has been done to elucidate this genetic code. So the code is basically a 3 nucleotide code. So 3 nucleotides are read. These are called triplet codes. So, now I said there are 4 kind of nitrogenous bases in the DNA or RNA. So if we say there are 3 nucleotides which forms a code, so there is a possibility of total 4 to the power 3, 64 codons. The codes are non-overlapping, so they are read in the units of 3. They are not overlapping. This is the example which is shown in this diagram here. Then the codes, they do not have any punctuation, so no sort of nucleotide is left. It's continuously read 3, and after and the next 3 set and the next 3 set. The codes are degenerate, so there is a redundancy in which the code is read, because if every code will have a unique amino acid, then you can imagine then you should have around 64 or amino acids or so, but that's not correct. So in natural occurring amino acids are 20. So that means that out of the 64 codes, some of the codes are going to code for more than 1 amino acid. So this is a kind of map you will come across, which will help you to read the codes. So you take the first letter from this column, second letter from this column, and the third letter from the other column, and you will see that for instance, this is UUU. It is going to code for a phenylalanine. There are certain codes which are not coding for any amino acid. These are called the stop codons. So basically at this place, if a machinery come across this stop codon, the translation is going to get stopped. Now the proteins, so once the information is read by these machinery, and the amino acid are going to be arranged, taking into reference how the codes are arranged on the transcript, you will have the proteins being formed. They also have a polarity. They have a free amino group at the N terminus and the free carboxyl group at the C terminus. These amino acids have a very basic, this is the basic structure of amino acids which forms these protein. They have a amino group, they have a carboxyl group. These carboxyl group and amino groups, they are involved in the peptide bond formation. There's a free hydrogen, there's a hydrogen atom, and there is a side group which is called the R group here. So there are different amino acids are also categorized depending upon the kind of side chain they have. So they are positively charged amino acid, negatively charged amino acid. There are polar amino acid, non-polar amino acid, aromatic amino acid. The property of that amino acid is defined by this R group. And that plays a very important role in the folding of these proteins, the way they behave in the physiological environment, the way they are going to interact with their binding partners. So, there is a very basic question that comes to one's mind, that the genetic information of the DNA is same in every cell, but we see a diversity in these cells. For instance, you have this erythrocyte, you have this epithelial cells, you have neurons. So they have same kind of genetic information. Then how come they look different, they behave different? So there has to be a sort of regulation or the way by which these genes are expressed in these different types of cells. So indeed there is. So they so basically on the chromosomes are present these genes. So genes is the basic physical unit of inheritance. So as I showed you about in the last slide, there are these pairs of chromosomes. So these are called homologous chromosome. One is from the male parent and one is from the female parent. So the location of a gene on this chromosome is called the gene locus. So there is a allele, there is a pair of allele which basically are alternative forms of a gene on a given locus. For instance, some people have blue eyes, some people have brown eyes. So basically the difference in the eye color they get from their parents, and depending upon which gene is dominant, that particular color the progeny is going to have. So these genes are around we know now with the Human Genome Project, there are around 20,000 genes which are present on the human chromosomes. So these genes are basically regulated, and there is it's quite a complex and equally beautiful process. There is a regulation at every level. It's a diverse topic. We don't have time to go into details, so I have summarized the major events which help in gene regulation at different levels. So at the DNA level, so when a DNA information in DNA is going to transcribe into RNA, so there is a lot of regulation that can happen, which is called the transcriptional regulation. So there are genetic elements. I briefly mentioned about promoters. So there are certain promoters, there are some inhibitory sequences. These are all cis transgenetic elements, though they play an important role as to which genes are going to be expressed at what time point in the cell life. There are epigenetic regulations. So I mentioned about histones. The DNA is packed around the histone. So there are modification of the histones that you can do, or which are basically some acetylation that can happen on histone, which basically the packing of the DNA is kind of decides which genes are accessible for transcription, or which are very tightly packed, they will not be accessible for transcription. So this is one way of regulating the gene expression at the transcription level. Then at the RNA level, you have these post-transcriptional regulations. So as I mentioned that you have the RNA that has some certain regulatory elements. You will have this intron elements present in between. So the splicing of this intron, the way it is spliced, that will also affect the way by which gene is expressed. This mRNA is supposed to be matured by the processing events. So the processing stability of this RNA, the regulatory RNAs, microRNAs, basically are the regulatory RNAs. These are also RNA molecules. They are not coding for any proteins, but they're going to go and bind to the complementary sequences on the mRNA, and they're going to decide the fate of that mRNA. Am I supposed to degrade this mRNA, or am I supposed to sequester the kind of information in this RNA by forming some complex structures so that the translation machinery is not able to read that RNA. Then at the protein level, you have some post-translation regulation, some modifications, some functional groups that you add to the protein, which can be reversible. For instance, phosphorylation is a reversible modification. You have phosphatase enzymes that will remove that phosphate. So depending upon at what time point that protein is supposed to function, maybe a phosphorylation is required for that. Then the kinases are the proteins that phosphorylate those protein, then they come and phosphorylate that particular amino acid. Now that phosphorylation is no longer required because the protein has to shut down the function, then the phosphatase will come and remove it. So it's a reversible modification. Then there are irreversible modifications. So they are ubiquitinylation pathways, which will basically go and mark a protein so that now no longer the function of that protein is required by the cell. So that attracts now the degradation machinery, and the protein is going to be degraded. So it's quite an amazing way how a simple looking cell is carrying out such complex cellular functions. Now coming to the experiment technique in the field. So the field has basically advanced a lot. So there are certain very basic techniques which were introduced in 50s or so, which has a huge impact on the modern techniques that we see now in the field. So I'm not going to go into too much detail into experimental techniques, just to give you an idea. So there is a lot of work going on, which basically adds a lot of data to the field. And this is what we are trying to address in this conference also, that how to manage that data, how to make a sense out of that data. So, and here I have just listed few important techniques. So we have now techniques to modify this gene. We can cut it at a precise location. We can change that particular gene by inserting some elements or removing some elements. We can now knock out certain genes or knock in certain genes into the animals where it's not naturally present. Then it's called the transgenics. We can sequence this gene. We can understand the exact sequence in which it is present. We can even sequence the proteins. We can overexpress these proteins. And then there are these microarrays work wherein we can actually understand how the genes is expressed in different cells. So just an overview of few techniques, a few basic techniques that forms the sort of basis of some modern advanced sort of techniques that you might come across in few lectures in next few days. So DNA sequencing was an important sort of step, a milestone in the field. So there are certain different sequencing techniques that were introduced, Maxim Gilbert, then Sanger sequencing. So Sanger sequencing is the one which got the maximum kind of acceptance. It's easy to use. You can use small amount of DNA and you can basically sequence it. This is the basic principle. So that Sanger sequencing techniques is also advanced nowadays. We do it very frequently. So this is the basic principle. You have this original DNA sequence. You PCR amplify this sequence with the help of this mixture of deoxynucleotide, DNTPs. And you also add in this reaction fluorescently labeled dideoxy, DD nucleotides, the dideoxy NTP. So dideoxy does not have that hydroxyl group on the third carbon. So that will basically restrict the formation of the phosphodiester bond, and the chain of DNA formation is kind of stopped. So different DDNTPs are labeled with different fluorescent tags, so that in a sort of automated system, you can read those fluorescent signals. You synthesize these DNA with the help of this mixture, and you'll run it onto this capillary gel electrophoresis. It's an automated system nowadays. So you have this laser beam, which will excite that fluorescent molecule, and you will read the fluorescence. And with the help of photomultiplied tube, you'll get certain graphs like this, which will tell you the sequence of that particular DNA. So which nucleotide got added, and then you can basically read the sequence of the DNA. So this DNA sequencing, nowadays we have this next generation sequencing. So it's a lot of added information. So basically it's a high throughput sequencing technique. So the inspiration is of course the previous sequencing techniques, but now we are doing a lot of advanced tricks and techniques so that we can sequence bigger genomes in a short interval of time. I'm not going into the details of next generation sequencing here. But then the gene expression analysis methods, there are several. This is one that I have just listed here. You can go back and maybe try to read more onto that. So DNA microarray is basically involves that you have these genes. For example, in this case, there are three genes A, B, C. This is their expression profile, the mRNA that is formed from these genes. What you do is you are extracting the RNA out of the cell, you are fluorescently labeling it, and you have this sort of microarray chip onto which this oligonucleotides are attached. Then you hybridize this fluorescent labeled RNA with this DNA microarray, and then you can read these signals. With the automated systems, you will be basically able to see that what is the expression profile of this RNA in the cell. And you will be able to see this. For instance, the B is not expressed so much, you have less RNA for B. So you will be able to see that it's, if you're going to detect, you will be detecting less expression level of gene B in this case. So DNA microarray is basically, this is another dimension, biomaterial in sort of engineering, where we come up with different chips which can basically help in the sequencing of, or the look at the expression profile of these genes. So it's very small chip. So millions of DNA strands are kind of built up on each location on this chip, and you tag your probes and they hybridize with this these DNA strands on the chip, and this is what you're basically reading in the DNA microarray. So I would like to come to the end of this talk by basically emphasizing once again that the basic molecular biology is contributing a lot to the field of data science. So these three major molecules that I have mentioned, DNA, RNA, proteins, they are contributing. There's a lot of data, or there is a lot of information present in these molecules, which is kind of read by these advanced techniques which are present in the field. So you have now several sub fields emerging from the field of biology. For instance, you have genome. So genome basically involves reading all the gene sequences. You have this copy number variations in the gene. You have single nucleotide polymorphisms. You're trying to understand you're trying to make sense what is going on. Why you have certain copy number variation between individuals. You have single nucleotide polymorphism. This kind of information is required because this will help you to go into the direction of personalized medicine. Then you have epigenome. Epigenome field basically look at the different modification, different individuals, the DNA methylations, histone modification, which has an impact on the gene regulation. Certain individuals are more susceptible to certain disease, the other they are immune. So this is basically because of this genetic variations. So this is an important emerging field contributing to a lot into the health of therapeutic, in the field of coming up with therapeutic techniques to and personalized medicine. Then there is transcriptome, RNA expression, RNA structure. There is a proteomics also nowadays. With the advancement in the mass spec techniques, you can look at the expression, the modifications, the present at the protein level also. So in terms of references, so you can refer to any basic biology book like Molecular Biology of the Cell from Alberts, Molecular Cell Biology from Lawdish and Freeman, the Cell and Molecular Biology, Karp and Pruitt, and there's a huge amount of literature present if you are interested in looking. And so basically you have to go on NCBI and you have to look at the latest publications if you want to have a more insight into the advanced information. So with this, I'd like to thank you for your attention, and I will be happy to take up any questions. Thank you so much.